So I'm going to talk about PDZ peptide binding, a less theoretical flavor. And I think, right, I've got the pointer. So I'm going to be talking about simplified free energy methods, Poisson-Boltzmann linear interaction energy, free energy ansatz, common class of simplified methods, as well as alchemical methods, both additive and polarizable. And this is work that was done mainly by these two students in my group. And I have an experimental collaborator, Ernie Fuentes at the University of Iowa, with his student, young Joe. And part of the work was done with help from Alex McRell and Benoit Roux for some of the polarizable parametrization aspects I'll get to. I think most people know these domains are protein-protein interaction domains. They have a certain level of specificity, a certain amount of promiscuity, and these things are essential for establishing correct interaction networks. We're interested in this particular protein, Chem1, and its PDZ domain. This is a protein that's overexpressed in some forms of cancer, as its name suggests, and it's actually a bona fide drug target. It's been targeted by at least one drug company, not very successfully, I believe. It has a typical 93 amino acids. It recognizes a few amino acids at the C-terminus of target proteins, typical of PDZ domains. Known targets include the SYNDICAN1 and CASP-R proteins. And importantly, it can bind the C-terminal peptide in isolation, so the peptide segment of the target can serve as a model ligand, but also as perhaps an inhibitor or bioreagent to affect the activity. And indeed, peptide binding to the PDZ domain of Chem1 has been shown to modulate its biological activity in cultures, cell cultures at least, and the activity, primary activity is to serve as a guanine exchange factor for the RAC1 signaling protein. So the peptide binds through a beta sheet augmentation mechanism, which you see here. The protein is blue, the peptide is orange. The C-terminus of the peptide is called position 0, and then amino acids are numbered backwards from there, the sort of typical PDZ convention, I believe. And here you have it in a bit more detail with the C-terminal position over here. The main specificity positions are the C-terminal one, and then position P2, two amino acids away, and they bind to each one to their own specificity pocket. Now, for us, this is primarily a model system. I'm not a biologist, and I'm not going to say a whole lot about PDZ biology, but this is a very nice model system. There's Chem1 in particular has a lot of data out there, much of it collected by Ernie Fuentes. We looked at a set of 51 protein peptide complexes, and when I say Chem1, I'm just referring to the PDZ domain, not the intact protein. And so we've used this to test and tune some free energy methods. The data set has some nice points. Of course, the ligand force field is well established since it's a peptide. Doesn't mean it's perfect, but it's the force field's out there. The data is relatively abundant with, for example, 28 peptides, 8 protein variants with bona fide affinity measurements, biophysical affinity measurements. A number of delta G values, including some that are actually just thresholds, lower bounds. These are negative controls, which are sequences down here in my list of peptides. There's also a combinatorial library of peptide binders that's shown here in the form of a logo, C-terminal position 0, and then the other amino acids going backwards, and you have the preferred amino acid types, which are indicated. There are some cons for the data set. Maybe the most difficult point is that the affinity range is not very large. Okay, we don't have a huge range here, only 2.2 kcals per mole. And also, the unbound state has a certain flexibility, a certain complexity, particularly the peptide. There's a limited number of X-ray complexes, so that many of the variants, most of the variants have to be modeled, usually 1
to 3 mutations away from an X-ray complex. It's not a tremendous homology modeling effort, obviously. So we played around quite a bit with this class of free energy methods with a free energy function, which is basically an ansatz of the form that's been used by a lot of people, quite a bit of work from the Jorgensen lab. This particular flavor includes a Poisson-Boltzmann binding free energy. It includes a van der Waals interaction energy and a buried surface term, all of these with their own adjustable coefficients. So one way to justify this ansatz, as most of you know, or many of you know, is to go back to these kind of free energy decomposition one obtains by following this type of binding path, where one would initially, in a thought experiment, turn off solute charges, then turn off dispersion interactions, bind your 2 cavities, and then reestablish dispersion and charges. And you can sort of think of each of these terms as a plausible ansatz for one or the other of these steps. And these are old ideas which help to motivate this type of object. So we model mutations with squirrel. Again, usually 1, 2, maybe 3 mutations away from an X-ray complex. We ran explicit solve and ND for relatively short periods, 75 nanoseconds on average. An amber force field, not exactly the most recent, but a reasonable force field. We suppress some rare unbinding events by some non-invasive semi-harmonic restraints on the peptide N-terminus. And the snapshots, as usual, get plugged into a Poisson-Boltzmann model where the solutes actually have a dielectric constant of 8. And this is a so-called single trajectory approach, where the unbound state is modeled by taking a bound snapshot and simply moving the partners apart. So fairly classical mix of ingredients. I would say the model is predictive. We fit these coefficients to 35 experimental delta G's, A, B, C. D doesn't, of course, really play a role in relative binding free energies. Coefficients come out with values not too different from some, although not all, similar studies of other systems. So there's some gross transferability, but not perhaps a huge amount of transferability. Here you've got experimental and computed relative binding free energies. This, of course, is perfect agreement. And then the outer lines here are 1 kcal errors. Over here, there's some millimolar binders to the right of this line, which we make no attempt to score. They're not part of the fit. And a single trajectory model is not going to be able to score those types of binders. Overall, there's a mean unsigned error of 0.4, 20% of the range. It's exactly the same level as a simple null model. There is a respectable Pearson correlation, I would say. Obviously, a lot bigger than the null model, which has no correlation, and not really any huge errors. So fair correlation, fairly small errors, better than the null model in the sense I mentioned. We did a lot of other things. We compared to a GB flavor, which is almost as good. We tried some other different flavors. And so hopefully what works, well, this kind of heuristic free energy ansatz works fairly well for the system. Single trajectory, we do have to suppress some rare fluctuations with relatively non-invasive restraints, exclude weakest millimolar binders, score separately some variants that have a distinctly different backbone confirmation. So there are a few things that can break, and one can actually build a very poor model if one really isn't too careful. What doesn't really work well, or at least certainly not work as well, plain MMPDSA, certainly not with a low solute dielectric constant. Overall, the model appears good enough to help design peptide binders as bioreagents, and we've done a high throughput design study which actually appeared in that same special issue of Chem Phys that was just mentioned. Otherwise, this work was published mainly in this paper here. So we moved on to alchemical simulations, which of course are going to make firmer predictions, at least that's our feeling, give additional insights and allow us to predict some new variants with a somewhat higher level of confidence in principle. So we have a alchemical Phet setup, which is corresponds to about 10,000 water molecules, a fairly simple vanilla windowing with dual topology, about 250 nanoseconds per mutation. We do almost exclusively point mutations, a couple of double mutations, around 250 nanoseconds per mutation. We've studied 20 peptide mutations and 2 protein mutations. So what some people now are referring to as edges. I think it's kind of a nice term. So this is sort of embarrassingly small compared to some of the data sets we saw this morning. There's enough here to keep a couple people busy for a while, I can tell you, though, nevertheless. And this does include all the experimental binding free energy changes for peptide single and double mutants. So there's a small set of affinities there. 10 new predictions, a few closed cycles, and a number of ionic mutations. Okay, so this is the kind of thing we get out. This is one of our, or this is starting to look like the graphs of mutations that we saw this morning. This is maybe a dozen of our mutations or edges. These are mutations at position 0. So remember, that's the C-terminal position shown here in the context of the syndican-1 peptide. And so here, for example, we're mutating the C-terminal residue from alanine to valine or methionine, say. We've got a computed value in black, an experimental value in blue. And for some of the variants, the binding is below the detection threshold, so we label as non-binders with a blue NB. And these red numbers are closure errors. This is one of the smallest in our full data set, and these are actually the 2 largest. Okay, so these are mutations over at position 2. You can see them in the context of the complex. Position 0 is here. Position 2 is here. We're going to be mutating 4 and also positions 1 and 3 in just a moment. Okay, so this is now the full set of calculations. So if you actually sat and scrutinized this data, you'd see there are 12 experimental free energy changes. There's 7 small errors, less than 0.5. So here, for example, or here. There are 4 that are around a kcal. Okay, so here we've got a kcal error, here as well. And then there's 1 large error. This is the single large error in our data set. So it turns out these larger errors all correspond to our ionic mutations, of which we have 7. And the 1 very large error is the mutation of this position 4 from glutamate into leucine, where the error is almost 3 kcals per mole. Okay, so we actually ruled out sampling issues, perhaps not perfectly, but did I think a pretty good job of empirically ruling those out here by doubling the amount of sampling, which led to no change in the computation. Here by tripling it, which led to a modest change. And we also simulated with the charm C36 force field, which adds another 200 nanoseconds per mutant and gives practically identical results for these 2. For example, here we go from 3.5 with amber to 3.6 with charm. Okay, so at this point, the obvious hypothesis is that there's a force field limitation here. And of course, one of the known limitations of additive force fields is that they overstabilize salt bridges in kind of solvent-poor environments, right? So if one looks at the syndican-1 peptide over here and the position 4, which is being mutated here, here, and here, the wild-type glutamate with the additive force field spends about half its time engaged in the salt bridge, either with an arginine or with a lysine, which is not present in the crystal structure. So our hypothesis is that this really is an additive force field issue, possibly due to salt bridge over stabilization. Okay, so we went ahead and set out to run some of these mutations, some of these ionic mutations with a polarizable force field, namely the Drude force field developed by Alex Macrel and Benoit Roux over the last decade or so. And many of you are familiar with this, and many of you know that the basic idea of this force field is that the normal particles carry a pseudoparticle which is attached by a spring, which is schematized here. So this would be, say, a carbon atom. And now we attach a pseudoparticle which carries part of the charge. So the charge gets distributed over the regular atom, the host atom, and the pseudoparticle. And there's a spring which has a certain stiffness. And the polarizability then shows up in the form of the kind of restrained motions of the Drude pseudoparticle subject to the spring. The Drude particle is light, and it has its own very cold thermostat. So it basically evolves adiabatically in response to local fields. Okay, and one can tune the charge separation to produce the correct polarizability or the desired polarizability. And so this force field is supported by more and more MD packages. Somebody mentioned that it's been ported into Amber now. I don't know if that's correct, but certainly Charm, Naden, Ramos, OpenMN, those are all published implementations. Dual topology FEP is mostly supported by NAMD. It's not fully supported, meaning that one has to do some things by hand at a kind of a low level. It's mostly supported. And the slowdown is about a factor of 4 compared to these additive force fields. So I'm not sure where Amoeba is at because there's been so much development of high performance Amoeba variants in a number of these packages. So I'm no longer really familiar with the timing, but certainly the Drude is about 4 times lower. So we applied this force field to the four mutations that had the larger error. And this is now, of course, a very small sample and a little bit anecdotal, but still, I think, quite interesting. And so the most striking thing that's happened is that our large error has really gone down to the level, kind of normal error level of about a kcal. So that was this mutation of glutamate into leucine here. We also have a very small closure, which kind of supports the level of sampling is not unreasonable. And overall, for these 4 mutations, which were basically our 4 largest errors, we have this, the errors I'm showing you here for these 4 transformations with Charm, Amber, and Drude. So the biggest effect was this large error vanishing, or at least going down quite a bit. However, if we look at the other ones, on average, they haven't really changed. Okay, so this one has gone down, this one has gone up. So over this very small data set, we've picked up a half a kcal error reduction, and we've eliminated one very large error. So I'd say that's quite a good result. Of course, it's a very small data set. I mean, this happens to be the first. Really don't have other applications to compare to, since this is the first time binding free energy differences have been computed alchemically with the Drude force field. So there's still a lot more to be done here. This came out recently. Okay, now one of the things we're actually, oh yes, so this is just a summary where you have the full data set, experimental, computed. The Drude variants are these circled dots. This is perfect agreement. These are kcal areas, and here you see this large area that's come down. And what is nice is that with the Drude force field now, the salt bridge occupation for that glutamate is down by a factor of 2. So a subtle but distinct change. Okay, now one of the things that was also motivating us to look at these polarizable force fields was the idea to look at side chain phosphorylation. Okay, so side chain phosphorylation is extremely important for regulating protein-protein interactions, and it turns out that Chem1 is sensitive to phosphorylation of the peptide at position Y1, which is here in my list of peptides, and right here in my complex. So phosphotyrosine was not part of the Drude force field, so we had to add it to the force field, which we've now done. We started out by looking at methyl phosphate. We did a full-blown Drude parameterization of methyl phosphate, which was also missing from the Drude force field. And we did this in collaboration with Alex McCrell and Benoit Hu, following the standard Drude parameterization procedure, which is an extension of the additive charm force field procedure, which perhaps more people are familiar with. You have to keep in mind that now an atom has a charge, but also a polarizability and a total damping factor. So there are 3 electrostatic parameters instead of 1 per atom. The philosophy of the parameterization is a little bit different from some other force fields. Obviously, one compares an extensive set of QM data to the force field model, electrostatic potential maps, but also these water interaction energies, just as with the additive charm force field. So this is methyl phosphate, and one considers interactions with a single water molecule along a donor or acceptor, hydrogen bond donor acceptor axis, such as this water here or this water over here. And so this is an example of a profile, an interaction energy profile with either the MM model or the QM model. This is for the monoanionic form of methyl phosphate. Okay, another type of QM property one looks at is dihedral torsion angles, and this is one example with the MM value and the QM value, the optimized MM parameterization. And this is the dihedral that corresponds to the methyl group swinging around sort of relative to the rest of the phosphate, and so on. So that's actually a fair amount of work to parameterize even this small model. One of the things we did, or perhaps I should say really the main test we did so far in the force field, is to compute magnesium phosphate binding free energies. And we considered both the monoanionic and the dianionic form of methyl phosphate, with both inner sphere and outer sphere binding modes, which are shown here. Okay, so this is the -2 complex, inner sphere complex, outer sphere, and so on. These are snapshots from some of the simulations. Now, okay, so to compute a binding free energy, we follow usual procedure, but which I've rarely seen in the form of a thermodynamic cycle. Which I think helps to clarify a few things. You'd be surprising how confused students and postdocs can get about how to really compute a binding free energy. So this is a kind of standard thought experiment where we start from a 1 molar ideal dilute standard mixture of methyl phosphate and magnesium. First thing one does, and one goes to an infinitely large or an infinitely low concentration. You think of large box limit, if you will. Then one decouples the magnesium charge, polarizability, Naden-Jones. One applies some kind of restraint potential that brings the two together. You recouple the magnesium, go back to the standard state concentration, and to finish up, you have to remove the restraint. And so you get your overall binding free energies. All the steps are done analytically. The only one that requires any calculation is the restraint step. It's the only one where there's a little bit of math. And of course, simulations show up in 2 places, the decoupling and the recoupling steps. And this role of the simulations is to compute the decoupling and recoupling free energies at some finite concentration, which is low enough so that one can then accurately extrapolate to the infinite dilute limit using simple analytical theories. That's the criterion for the box size. You don't have to be at the large box limit, but you have to be big enough you can accurately extrapolate with a simple analytical theory. Okay, so having done all that, we get binding free energies. Now I should mention that for this dianionic phosphate magnesium complex, we did it with an additive force field a number of years ago. Error is about 40 kcals. Okay, and that's going to be true, I think, for any additive force field. Here, we have experimental number for the -1 form is minus 1.7. So sorry, Jay, it was not 2.1. It's minus 1.7. And computationally, this is a number not for methyl phosphate, but for pure mononegative phosphate, inorganic phosphate. There's no number I could find for the methyl form, but I think they're close enough so it's a valid comparison, if not a completely quantitative comparison. So we're very, very close, and the binding is, free energy is computed to be 0 for the inner sphere mode and minus 2.1 for the outer sphere mode. If you go to the dinegative form, the experimental value for inorganic phosphate is minus 2.7. We get minus 2.8, again, almost entirely produced by the outer sphere binding. And I should also mention that a few years ago we did the mononegative form with amoeba, and we also found that we had actually very similar numbers, and also very good agreement with the experimental affinity. So the outer sphere binding has been found now by 2 polarizable force fields for the -1 form. Perhaps the -2 form maybe needs a bit more verification. But, so the student, Francesco Villa, took him several months to parameterize methyl phosphate, but then he went on and did phosphotyrosine, and it only took a few days. So once you're up to speed, these things can be done in a relatively short time frame. So phosphotyrosine is underway, and if the meeting had been a week later, I could have shown you delta delta G values. I can also mention that we did a thorough study of the phosphotyrosine variant binding to Chem1 with an additive force field. And again, the results are absolutely terrible. It's a complicated problem to study. There are 2 protonation states, multiple conformations, and so on. So I'm not sure how well Drude is going to do. But if it can do magnesium, I'm pretty sure it can do this. So this has been a relatively academic presentation for this drug-oriented conference. Of course, hopefully it's sufficiently interesting also for non-academics. Of course, some people will say that French scientists are too theory-oriented and maybe not. And there are actually some American thinkers who have commented on this famously, I would say. In French, we would say, ça ne s'invente pas. And so I'm not going to summarize my talk. I'm simply going to acknowledge once again my coworkers and a very, very good set of collaborators, including some things underway, which I didn't talk about, which are hopefully going to be fruitful with Mike and Jacob, who are here. And thank you all for your attention.
Questions?
I'm the one who's jet lagged.
I might have missed it, but how are the experimental energies determined for the -1 and -2 charge phosphate?
Good question. There are a bunch of experimental techniques which I've looked into a couple years back. I believe there are chemical shift measurements with phosphorus NMR as one technique. There's some resin absorption techniques where one can detect okay, I'd have to mull over it a bit more. There are 4 or 5 different techniques.
Yeah, maybe you can let me know after.
No. These are not super old measurements. Different people have done the -1 and the -2 forms, and I think one of them is in the 80s. Alberti did one of the forms. I would say mid-80s.
Other questions?
There's a shortage of thermodynamic data for these phosphate compounds, including GTP, ATP, amazingly sparse data sets.
So how does the polarizable force field dampen the, you said that pairwise ones in a non-polar environment overestimate the stabilization.
Right, which you can imagine, right? I mean,
Right.
that's what you would expect.
So how does the polarization, what's happening there that's
Well, there's
damping
less,
that?
there's less intrinsic charge separation, and the charge separation develops when you approach the magnesium and the phosphate. I mean, you know, there are people here who, we've done a fair amount of work with polarizable force fields, but obviously, you know, right? But I mean, that's the physics, right? You don't develop the induced dipoles until they're induced, right? That's.
Any other questions for Tom? Okay, let's thank him again and all our speakers from this afternoon.